caused by parasitic protozoans and is one of the leading causes of death in children. In 2012, 207 million cases and over 600,000 deaths were reported to the CDC, with over 90% of those deaths occurring in Africa. Persistent malaria infection is common in endemic areas such as Africa. It is caused by the lack of immune activation despite the presence of parasites. So the host and the parasite can coexist, and the host may be asymptomatic. Possible mechanism for this phenomenon is lymphocyte exhaustion, in which the lymphocyte responses such as cytokine proliferation and production is inhibited. We've created a model of persistent malaria infection in mice by using Plasmodium jabati, which is a strain of malaria that infects rodents, including mice. But to explain how we created this model, we must first understand how white blood cells clear the infection in the first place. So CD4 positive T cells are white blood cells that release a pro-inflammatory cytokine, interferon gamma, which activates B cells to release antigens, antibodies in response to antigens, which then clear the infection. So we used two components to create persistent malaria infection. One is using interferon gamma receptor knockout mice, which we just buy as a certain genotype from a company, and which blocks the interferon gamma signaling pathway. And the second component consists of using a GK1.5 depleting antibody, which temporarily eliminates the CD4 positive population. Now, each of these in isolation has an effect on the infection, but only in combination do these cause a persistent malaria infection. This is a graph depicting parasitemia levels in persistent versus acutely infected mice. So as you can see by the red line, the mice treated with the GK1.5 antibody show high levels and consistent levels of parasitemia as opposed to the control, so the black line, which is the LTF2 treated mice, which for our purposes here do not bite to anything of interest. And as you can see, on day 40, high parasitemia is established. So all the data preceding this will have been taken after this high parasitemia level has been established. So what are masks and why are we interested in them? Masks are what we are calling malaria-associated suppressor cells, although this is not an official term. This is what we're choosing to call them at the moment. And they are a subset of monocytes that present surface markers traditionally found on non-classical and dendritic cells. We identify masks using non-classical monocyte markers because they are similar, although not identical to these non-classicals. And we're interested in them because we're looking at them as a possible mechanism of immune suppression during persistent malaria infection. And we see that they upregulate programmed death form ligands, which cause cell death in the cells with the PD-1 receptors, such as T cells and B cells, which leads to inhibition of the immune response. This is a snapshot of the leukocyte count on day 100 of two different types of mice. So on the right side here, you see the cleared mouse. So the mice have had infection, were infected, but they cleared the infection. And as you can see by the red sliver, the masks only make up a small proportion of the total leukocyte count. Now on the left then you see the persistently infected mice where the masks make up a significantly larger proportion of the leukocyte count. So it's interesting because it's expanded in comparison to the cleared mice. We also see T cell suppression during persistent infection. So initially after treating with the GK1.5 antibody, which depletes CD4 positive T cells, over time the CD4 positive T cells do repopulate. However, fewer are antigen-experienced CD4 positive T cells. That is to say, specific for the malaria. So as on the left-hand side there, over there, you see the yellow line. So this antigen-experienced CD4 positive T cells do not repopulate nearly as much or as well. And we know that these CD4 positive T cells are specific for the malaria infection because these mice have been in a controlled environment and were only exposed to malarial parasites. As mentioned before, the masks upregulate PD-1 ligands, so PD-L1 and PD-L2. infections, and as you can see by the large red peaks there, they have more expression of PD-L1 and PD-L2 in comparison to the non-classicals and the naive mice. So how does the body go about increasing the population of cells? One way is to increase the input, that is to say, proliferation. Another way is to decrease the output, or decrease cell death. And the third is to traffic from other compartments or aggregate. So to bring in the cells from other areas of the body into one area. Our lab has been focused on decreasing output. And one reason why we focus on this is due to data that we also collected, which shows that there is less non-classical cell proliferation shown in persistently infected mice in comparison to naive mice. So Ki67 is a nuclear protein essential for proliferation, and as such is a marker for cell proliferation. As you can see, the non-classical and the persistently infected mice show a lower percentage of Ki67 expression than the non-classicals of the naive mice. On the left side over there. So there are several methods of cell death. Apoptosis is one of them, which is naturally occurring cell suicide, which can occur due to removal of the cells from live tissue or organs, change in temperatures from body temperature to extremely cold or to extremely hot, induced immune responses, etc. Necrosis due to external damage, so due to traumatic injuries, toxins, infections, autophagy, self-digestion, which causes degradation of dysfunctional or necessary cellular components, and there is many more, such as characterizing, inflammation, mitotic catastrophe, mitosis, entosis, division by another cell. Our lab chose to focus on apoptosis because it is one of the most common methods of cell death, and we do not have evidence to suggest that the other methods are occurring at this time. So to characterize apoptosis, I have to follow a couple steps, and I will break them down as I go. First is to recover the blood from the infected and naive mice, then to stain these subset of cells using various assays for apoptosis, and then to use flow cytometry to identify the types of cells that we've acquired. So we recover the blood by submandibular blood collection, which is to use the facial vein in the mice, so on their cheeks right there. And we collect about 50 to 150 microliters of blood. Then we stain the masks using various assays for apoptosis. One includes annexin 5, which stands for phosphatidylserine exposure. So in viable cells, phosphatidylserine is a component of the cellular membrane that is in the inner leaflet. However, in apoptotic cells, the phosphatidylserine is flipped to the outer leaflet so that the annexin 5 can bind to it. Rubidium iodide stains for DNA fragmentation, and in viable or live cells, it is unable to permeate the cellular membrane to stain the DNA fragments. However, in apoptotic cells, the membrane is compromised, so the DNA is able to permeate the membrane and stain the DNA fragments. The same principle applies for 7-AD, which stains for DNA fragmentation, and there's also mitochondrial staining, which stains for mitochondrial changes in potential. Finally, we use flow cytometry to identify these cells. I won't go into much detail, but basically what happens here is that the cells are put into a machine, a single file. Lasers are shot at the cells, and the cells fluoresce depending on the stains and the bodies that you use. And the machine collects information and kind of outputs the type of cell that it is. And this is an example of the kind of titrations I had to perform in order to get the optimal dilutions to separate clearly the positive populations from the negative populations, that is to say, distinguish between the lag cells and the apoptotic cells. And my results have shown suggest that there's decreased mass apoptosis in infected versus naive mice. So the top two figures are two separate mice. So, first, all these mice are the same genotype. So they're the interferon gamma receptor knockout mice. And the top two are showing two different types, two separate mice. used, if you see those squares, I used their circling different populations, and the top one is circling the NXN5 and PM-iodine positive populations in apoptotic cells, and the bottom one is circling the NXN5 positive and the PI negative cells. And this kind of L-shape, reverse L-shape is characteristic of apoptotic cells because they undergo a specific process in which the membrane flips and then degrades. And I applied those gates to the infected cells because the populations weren't as clear there. And these are my results. As I stated before, the mask apoptosis in the persistently infected mice seems to be less as compared to the naive mice. So you can see the NXN5 positive and the PI positive levels are lower in the persistently infected mice. Now this is suggestive of expansion of masks through decreased cell death. However, my population, my sample size was very small, so this is by no means totally conclusive. So in the future, I would like to expand the experiment to increase my sample size, reproduce findings, that is to say, conduct more trials, and use different assays to corroborate my results. And in the future, also, it would be interesting to adoptively transfer these masks into mice of different levels of infections, such as naive, acutely infected, and then persistently infected, to determine whether the signals of survival are intrinsic or extrinsic to the cell. I'd like to thank Dr. Charlie Kim